All right, thank you all for joining us today for this installment of the Cape Perpetua Speaker Series. We're going to be hearing from Will Fennie, and he's going to present to us about the vulnerable early life stages of black rockfish in changing ocean conditions. And next week, we will be hearing from Mary Coolidge on the about From Desert to Coast, the Case for Dark Skies. And a little bit about the Cape Perpetua Collaborative. My name is Tara Du Bois, and I'm the Communications Coordinator, and it's a pleasure to coordinate the series. And the Collaborative's vision is to foster conservation and collaboration within local communities for scientific exchange, management, awareness, and stewardship from the land to the sea and around the Cape Perpetua Marine Reserve. And our three guiding principles are community engagement, leveraging resources, and engaging in partnerships. And as you can see at the bottom here, these are our founding partners. We organized in 2017, and our founding partners include federal agencies, state agencies, some local nonprofits, as well as the Confederated Tribes of Coos, Lower Umpqua, and Siuslaw Indians. And our focus is on the Cape Perpetua Marine Reserve, and in addition to the marine protected areas to the north and south, there's some form of protected waters that stretch between Yachats and Florence. Cape Perpetua is the largest of 5 marine reserves in Oregon. And today's fun fact, I just wanted to share about what do Oregonians love about the coast. A recent study out of PSU looks at how Oregonians value different places on the coast. This was an open survey. There was a map of the coast with 15 potential values, and online participants were asked to place pins, value pins at these locations. And there were 318 online participants and 9,700 pins placed in coastal locations. And so they also looked at specifically at the Oregon Marine Reserves, and placed out the top 3 values for each marine reserve. And for Cape Perpetua Marine Reserve, the top 3 values were aesthetic, biodiversity, and beach recreation. And you can find out more information about this study if you head to the OregonMarineReserves.com website and go to Reserve News, and this will be the first post you'll see on that page. And they have a link where you can download this study. We do host a variety of community science projects in the area. Some of them are seasonal, some of them are monthly. And we also host a Young Scientist webinar series on the 2nd Tuesday of the month, October through April. And our next one is coming up this Tuesday at 5:30 p.m. And it's going to be on understanding the relationship between commercial fishing and feeding, an ecology of the Dungeness crab. And you can find out more information about all of these events at our website at capeperpetuacollaborative.org. And I also encourage you to connect with us on our Facebook page and our YouTube channel. And now to introduce our speaker, Will Fennie. He recently earned his PhD in the Plankton Ecology Lab at Oregon State University. His research examines the influence of oceanographic conditions on the early life stages of black rockfish. He previously examined the influence of ocean acidification on the behavior and swimming ability of rockfish during his master's at Moss Landing Marine Laboratories and completed a BS in Biological Sciences at University of California at Davis. Will is fascinated by rockfish, and when he's not studying them, he likes to free dive and take pictures. And with that, I'd like to turn it over to Will. He is joining us today from sunny San Diego. Welcome, Will. Thank you for being here with us today.
Good morning, and thank you for having me.
And while Will's pulling up his PowerPoint, I just want to let everybody know we will do Q&A at the end of his presentation. So if questions come up throughout, feel free to add them to the Q&A or the chat, and we will answer those at the end. Take it over, Will.
Well, thank you, Tara, very much for inviting me to talk to this group. I'm really happy to be here today, and it's a great opportunity for me to reconnect with people from coastal Oregon, a place that's very near and dear to my heart. So I'm going to jump in and start talking to you all about the vulnerable early life stages of black rockfish in changing ocean conditions. Now, fish and fish populations are really important both ecologically and economically. Forage fish species such as sardines and anchovies, as well as young life stages of rockfish, are important energy conduits linking primary productivity in the plankton to higher trophic levels. And this photo on the left here shows a swarm of recently settled juvenile rockfish in Pacific Northwest. And they create a smorgasbord for a variety of marine species. And because rockfish eat really low on the trophic level, at least in this early life stage, they bring that energy up to larger predators such as seabirds, large benthic, recreational and commercially important fishes, as well as marine mammals. So they're a super important link in their ecosystem. Additionally, fish populations are important sources of income to coastal economies. They can provide opportunities for tourism, as well as commercial and recreational fisheries. And in Oregon, I think about 7,000 jobs are created by the fishing industry, and it's a major driver of coastal economies. Because of that, there's been a lot of work trying to understand why fish populations can fluctuate so much from one year to the next. And this is a centuries-old question. In the early 1900s, a group of fishery scientists in Europe tried to understand why Atlantic cod and herring populations could vary by almost tenfold from one year to the next. And they spent a lot of time and energy trying to ask this question. And during the development of their research program, this really new technique became really important to helping them. And that was the advent of fish aging. So early on, scientists were able to take fish scales off of herring, and eventually they developed techniques to use fish ear bones, or their otoliths, to age fish in both how old they are in years, and then later they realized they could figure out how old they were in days during their early life stages. And this completely changed the game for figuring out how fish populations fluctuate. So this figure on the right here demonstrates how aging fish allowed fishery scientists to track populations through time. And what we have here on the top is the age of fish. And then we have the year that they were studying these populations at the right of these figures. And I want you to think about the task in front of these fishery scientists. So they had to go in and look at a population of fish and try and understand why there are a certain number of individuals in a given year and why that would change from one year to the next. So think about if you were trying to do the same thing with a school of people, perhaps like a high school class, and you couldn't identify the ages or the classes of students. So you might have, I don't know what a typical high school in Oregon is, maybe 400 or 500 people. And you don't know how many are freshmen, sophomores, juniors, or seniors. But then this technique comes around and you can actually age them or ask them how old they are, and you'll see something really amazing happening. So this first year, we see a really large bump in the 4-year-old age class relative to ages 5, 6, 7, and so on. And so think of this as a huge freshman class coming into a high school and completely changing the population size of the school. And then if we go to the next year, you'll see that now there's a really big sophomore class that transitioned from their freshman to sophomore year, and a decrease in the number of individuals from the other year classes. So this really points to the importance of a particular year class in determining the size of a population. And what this led to was the understanding that survival during fish early life stages was actually a major driver of variability in adult populations. And this led to the development of the critical period hypothesis, which suggests that in the first few days of life, the ability of fish to find food and to experience currents that retain them in favorable habitat is really important for determining survival during their early life stages, and translates into how a population will fluctuate across time. And this discovery and hypothesis fueled fisheries oceanography for the next century. And what this looks like, I'll try and illustrate here. So we have fish age in days on the x-axis and the relative abundance on the y-axis. And female fish are highly productive, with several species producing hundreds of thousands to millions of young each year. And typically survival is really low during these early life stages. Fish are just millimeters long. They're vulnerable to a variety of predators. Basically anything that can fit them in their mouth will. And then they have to find food during their first few days of life to prevent starvation. And in years where conditions are unfavorable, such as there's low food availability and potentially currents are moving them outside of favorable habitat, very few fish survive from the day that they were born to the day till a couple of days of age. And when there's really low survival during the first few days of life, this translates to few fish surviving to recruit to the adult population. However, in years when conditions are more favorable, such as there's much more food available and ocean conditions kind of keep the larval fish in close contact with their prey source, survival during the first few days of life can be much higher. And this translates into a lot of fish surviving throughout their early life and bolstering the adult population. And over the last century, there's been a lot of work identifying the different sources of mortality that affect survival during these really vulnerable life stages. So this figure has the same axes as before and shows how a lot of the mortality that occurs during a fish early life happens during the youngest life stages. As fish move into their larval and juvenile stage, they become a little bit more resistant to mortality. And another discovery was that predation occurs across all life stages and is thought to be the major source of mortality, determining population sizes from one year to the next. So there's been a lot of work trying to understand if there are certain traits young fish possess that would make them more resistant to predation and would help us predict survival during these vulnerable life stages and allow us to understand population fluctuations. And this led to the development of the growth mortality hypotheses. They are very creatively named. So there's the bigger is better hypothesis, which suggests that for fish of a given age, larger individuals are more likely to survive than smaller individuals. And this is because fewer predators can fit them in their mouth if they're bigger, and also larger fish can swim faster. So this allows them to avoid predators and find prey more easily. So it's a big advantage. Next is the growth rate hypothesis, which suggests that individuals that are growing faster at the same age as other individuals are more likely to survive because they'll achieve a larger size more rapidly, and this increases their survival. And finally, there's the stage duration hypothesis, which suggests that the more rapidly fish develop and metamorphose from their larval to a juvenile stage, the more likely they are to survive because they spend less time in the really vulnerable early life stages. And these hypotheses have been tested in a variety of species and systems, but mainly in the Atlantic and in the tropics, with few studies along the West Coast of the United States. And that's what I wanted to do, is apply these hypotheses to an important group of fishes that are found commonly along the West Coast. And for those of you who are not completely enamored with rockfish, let me sell you on them a little bit. So there's over 100 species of rockfish found worldwide, 70 of which are found along the West Coast of the United States, and about 40 of which can be caught off the coast of Oregon. Besides being beautiful and delicious, they are also characterized by being really long-lived, with many species living to be several decades old and several living to be well over 100 years old. They're also slow-growing and slow to mature, which makes them highly vulnerable to overfishing. And indeed, these fish populations have had some struggles with fishing pressure through time. So this figure shows years on the x-axis and essentially abundance of the fish population on the y-axis. And this is data provided by NOAA's FRAM survey. And I just want you to think about this 100% line is indicative of what the fish population was like prior to fishing exploitation. So if the population, if the lines are close to this 100% line, that's when their populations are really high and have not been impacted by fishing. And when they get below this 25% threshold, that indicates that their populations have declined dramatically and are in danger of collapsing because of really heavy fishing pressure. And what this figure shows is that a variety of rockfish species commercially caught along the West Coast declined dramatically during the 1980s into the 2000s. And fortunately, really strong management actions were implemented in the early 2000s to protect these species and allow them to recover from this heavy fishing pressure. And there's been a lot of work trying to understand what's happening in the early life history of rockfish, because this is the period in their life that's going to determine how quickly these overfished populations will recover and allow us to better manage these species in the future, especially as climate change continues to progress. Now, rockfish, like many benthic species, have a biphasic life cycle. So adults are largely sedentary. They find a rocky reef that they hang out on, and they don't move a whole lot. But they do have a dispersive early life stage where they give birth to larvae that are advected offshore and hang out in the plankton for a couple months before metamorphosing into a juvenile life stage. And these juveniles start out offshore, and then for all the nearshore species, they actually have to make movements to nearshore habitats, and they will settle to habitats like kelp canopy or eelgrass beds or nearshore rocky reefs. And if they survive the first couple of years of their life, they'll then become adults and recruit to the adult population. Now, survival during their early life is complicated by the fact that they live in the California current. And this is a really dynamic oceanographic habitat. Currently in Oregon, you're in the winter stormy and downwelling season where there's lots of strong winds coming out of the south, big waves that mix up the water column. And this is right around the time that many species of rockfish actually give birth to their larvae. And then in a few weeks, if you hang in there, winter shifts into the spring and summer upwelling season where the valley starts to warm up, and this causes really strong northwest winds to blow along the coastline. And these winds, coupled with the rotation of the earth, will drag surface waters offshore and cause cold, nutrient-rich, and highly productive water to be upwelled into the nearshore habitats. And this really increases the productivity of the system and it's thought to be a really important driver of survival for rockfish, as well as many species that inhabit the California current. This figure on the left is a satellite image showing sea surface temperature along the west coast of the United States. And this band of green along the coast indicates really cold water that has been upwelled and indicates really high productivity during the spring and summer months coastwide. And the strength of upwelling is modified by two major oceanographic modes. You may be familiar with some of them. So there's the El Nino Southern Oscillation Index, which occurs on a 3 to 7 year time scale. And currently we're in La Nina, which creates lots of storms in winter and lots of rain up in Oregon. And in terms of the ocean, it leads to really cool water along the coast and is indicative of strong upwelling and high productivity. In contrast, the other phase is the El Nino. And this is where it gets much rainier in California. Water is a lot warmer along the coast. Upwelling is reduced, and so is productivity. So these happen on a couple year timescales. And in addition, there's also the Pacific Decadal Oscillation, which happens on much longer timescales of 10 to 20 years, although there's some evidence that that's starting to speed up a little bit. And similarly, there's a negative cool phase where conditions are colder along the west coast of the United States, and this leads to stronger upwelling and higher productivity. And then this can switch for a decade or so, and there'll be much warmer water along the coast, and this leads to decreased upwelling and lower productivity. So there's a lot of work trying to understand how the strength of upwelling affects survival of rockfish and whether or not that's a good predictor of recruitment or survival during their early life. And several efforts to look at rockfish recruitment variability or survival indicated that upwelling was a pretty good predictor, and that in years where there was really high survival of rockfish tended to be years of stronger upwelling, and in years with much lower upwelling led to lower rockfish survival. So for a while, scientists thought they had a pretty good handle on rockfish early survival. And then the large marine heat wave impacted the California current. And this totally threw our understanding of how upwelling impacts survival of rockfish. So if you remember, this started in the summer or fall of 2014 and really impacted the West Coast in 2015 and 2016. We saw a variety of new species from more tropical latitudes coming up to the coast. I think pelagic red crabs are really popular, are really common on Oregon beaches, and they typically don't extend north of Mexico in normal years. In addition, there were also pyrosomes that were found throughout the West Coast. And this was a species that is typically found in tropical latitudes and was indicative of a complete change in the biological community during the heat wave. And what this showed is that we really lack a mechanistic understanding of rockfish settlement variability and survival during their early life stages. And so my PhD work was trying to use rockfish early life history traits and the growth mortality hypotheses to try and get a better handle on why survival of their early life stages varied so much from one year to the next, and to figure out if we could use that information to predict how their populations would change in the future. So what I did was I collected settlement stage juvenile black rockfish from 2013 to 2019. And this included the large marine heat wave years. And if I have time today, I'll talk to you a little bit about another chapter where I looked at predation of salmon on rockfish and whether or not their early life history traits were a good predictor of survival in a direct predator encounter. We'll see. I got to get moving if that's going to happen. So to think about how rockfish early life history traits and especially growth may be important to help understand how their populations fluctuate in novel oceanographic conditions, we had to collect a bunch of fish. And just quickly, a reminder about who and what black rockfish are. They are one of the most commonly caught species on the Oregon coast in the recreational fishery. They can live to be about 50 years old. They're found pretty much in any rocky reef within, the 100-meter or 100 fathom depth limit. They are delicious. They're favorite food of spear fishermen and hook and line fishermen, and their juveniles settle to nearshore habitats during May to July. And the hypotheses for this project were that their larval growth rates will vary with environmental conditions. And this is because fish are largely cold-blooded, so their metabolism is directly linked to the temperature that they experience. So if their environment's we thought would be a major predictor of what their early life history traits would be like. And then we thought that oceanographic conditions leading to faster growth rates will correspond to years of stronger black rockfish settlement. To do this, we had to partner up with this really awesome collaboration that's called the SMRF program. And it's comprised of scientists from Oregon State University and headed by Dr. Kirsten Growred-Colvert. And there's a ton of support from the Oregon Marine Reserves Program and Kristen Don and Dr. Lindsay Aylesworth. And then we really rely on partnering with the Oregon Coast Aquarium and Jim Burke, Jenna Kloss, and Doug Batson for help with our sampling for this. And a SMRF is not only is it a little blue creature, but it's also a standard monitoring unit for the recruitment of fishes. And my former lab mate Danny here is holding a SMRF, and it's a cylindrical tube of garden fence mesh that's stuffed with more fencing that mimics kelp canopy habitat that many juvenile fish use to identify as suitable settlement habitat. And we're able to free dive on these Smurfs that are hooked onto moorings just outside of the Otter Rock Marine Reserve and also in Cape Foulweather. And we engulf them in these nets, we unhook them from the moorings, we bring them up onto the boat and shake them out. And then we can identify all the fish species that we collect and do some pretty cool analyses with them. Now, because there's so many different species of rockfish that settle to the Smurfs, we have to be able to identify them to species. And this took a lot of time to figure out the difference between juvenile black rockfish and juvenile yellowtail rockfish, because they have very similar characteristics. But once we got through that hurdle, we began to dissect their otoliths, or their ear stones, out of their heads and use these to find some really valuable information. So you can see these little bones hanging out right here in the fish's cranium, and they're important for both hearing and 3D spatial orientation. And they also lay down layers of material on a daily basis. And it may not look like it from this image of a whole otolith, but when you polish them down into a really thin wafer, they provide a wealth of information. So here's a pretty well polished black rockfish otolith. And you may notice they look a lot like cross sections of a tree or a tree cookie, with these concentric rings extending from the center all the way out to the edge. And these rings indicate material being laid down each day of a fish's life. So we can count these rings to get an exact age of the fish in days. And if we zoom in on the core here a little bit, this first dark ring indicates when the fish was extruded by its mother. So this indicates its first day of life. And if we count each band all the way out to the edge, we can get an exact reading on how old a fish is. And then if we measure the distance between each ring, we can figure out how quickly they were going on any given day of their life. We can also identify when the otolith starts to create secondary growth primordia. And this is basically an indication of when the fish starts to undergo metamorphosis. The otolith starts to grow in weird directions. And when we line that up with the ring that corresponds to when this structure formed, we can figure out exactly when they started to metamorphose and determine how quickly they're developing. And then we have to couple that early life history information with oceanographic information. So here I've compiled monthly mean values of a variety of oceanographic indices. And we use these to match up to the exact timing of when the fish were born to figure out what they are experiencing during their early development and how that might impact their growth and their survival. So here we've collected information on the Pacific Decadal Oscillation. Remember, this is the longer term fluctuation in temperature and upwelling strength. We have a measure of the Ocean Nino Index, which is a measure of the shorter term fluctuations in temperature and productivity. And then there's the North Pacific Gyre Oscillation, which is a direct measure of the vertical transport of nutrients, which indicates how productivity was enhanced by upwelling. And we're going to walk through what the conditions were like in each of the years. So here I have a satellite sea surface temperature map that corresponds with the timing that we collected the rockfish and the environmental information that we're using. So 2013 was also a La Nina. It meant that it was very cold along the coast of Oregon. There was a negative phase of the PDO, a negative phase of the El Nino, and there was high productivity due to upwelling. As we move into 2014, you can see the formation of the warm blob in the Northeast Pacific. It hasn't yet really impacted the coastline, but it's coming. And this is indicated by the switch to the positive phase of the PDO, a slight change in the Ocean Nino Index, and a decrease in the transport of nutrients. And then as that heat wave hits the coastline, we see the PDO and ONI values really skyrocket and the NPGO decrease. And this just shows that the temperature has totally warmed up and that it has shut down upwelling. And then we see a change in 2016 where there's, a still strong PDO and then a really strong El Nino. And this is indicative of dramatically different conditions that are still warm, but slightly different than the first couple of years of the heat wave. And then the heat wave starts to diminish and conditions start to return to their cooler phase. But by 2019, there was another formation of a warm blob and fears of another drastic heat wave that didn't hit the coast nearly as hard as the 2014 and 2016 heat wave. We also tracked some biological indicators, including the northern and southern copepod biomass anomalies. And what these do is they give us an idea of what the plankton community was like and what the diets of these fish might have been like during this time period. Northern copepods are large in size. They come down from the Northern California current, and they're really lipid rich, which makes them a really high quality prey source. There's also the southern copepods, which come from the tropics. They're much smaller in size. They have fewer lipids and are thought to be an inferior prey source. And their patterns in abundance in the California current are mirror images of each other. So the northern copepods are really abundant in the cool years prior to the heat wave. Then they crash during the heat wave. They're just very rare. And then they start to come back after the heat wave dissipates. Southern copepods show the exact opposite. They're rare prior to the heat wave, much more common during the heat wave, and then they diminish as the heat wave receded. We also tracked a local measure of sea surface temperature at the Stonewall Bank NOAA buoy, and this just shows how temperature fluctuated during their early life stages more directly. And we also measured coastal upwelling transport index. So now that we're armed with a battery of oceanographic variables and we've extracted information from rockfish otoliths, we need to look at the patterns of their early life history traits and then compare them with oceanographic conditions. So here are a suite of rockfish real life history traits. So we have their mean larval growth. And this was acquired by measuring the distance between consecutive bands in their otoliths for each day of life for all individuals for a given year. So here on the x-axis, we have fish age in days and their mean daily growth. And what I want to draw your attention to is that we found kind of three different groups based on their growth rates. There's the really fast growers that are from 2014 and 2015. There's the moderately fast growers in 2016 and 2019. And then there's the slow growers in 2013, 17, and 18. And when we take the average of their growth rate across their larval stage, we get this figure, which shows slow growth in 2013 and 2017 and 18, fast growth in 2014 and 15, and moderate growth in 2016 and 2019. And their growth rates were strongly correlated with their age at settlement. So when fish were growing really slowly, it took them longer to settle. And when they were growing very quickly, they settled at very young ages. And this also was reflected in their development rate. So here we have the relationship between their larval growth rate and their age at metamorphosis. Remember, that's when their otoliths start to change shape, indicating that they are now transitioning to their juvenile life stage. And in 2013 and 18, when fish were growing really slowly, it took them 65 days to start undergoing metamorphosis. But in years when they were growing really quickly, they were metamorphosing almost 10 days earlier. So this is a really drastic change in their developmental rate. And then we looked at how this affected their size at settlement. And this was somewhat surprising. It showed that for most years, they didn't settle until they had reached kind of a common size. So this meant that when they were growing faster, they could settle earlier at a younger age, but they were still going to be the same size as individuals that were growing slower, with the exception of 2015, where they were just really, really small. And this means that they grew quickly and settled at a really young life stage. And finally, we wanted to see how this translated to survival. So we found that survival was really low in 2013 and 2015, moderate in 2014, 17, and 18, and really high in 2016 and 2019. So next we had to correlate these early life history traits, and especially their growth rate, with the environmental conditions they are experiencing. And this figure shows the results from a partial least squares regression analysis. And I don't expect you to know what that means. All I want you to look at is the direction of this arrow indicating their larval growth rate. And the two arrows that most closely follow that are sea surface temperature and the Pacific Decadal Oscillation. And these two variables explain almost 70% of the variability in rockfish growth rate. So this indicates that really local and basin scale temperature signatures are the best predictors of how quickly rockfish are going to grow. And here I've plotted their growth rate versus essentially temperature. And this largely followed what we expected. Because rockfish are cold-blooded, in years when temperature is warmer, they're going to grow faster. And this is what we found, is that their growth rates increased in years with warmer water conditions. Next, we looked at how oceanographic conditions affected survival during their early life stages. And we were pretty surprised to find that their settlement rate, or their survival to settlement, didn't match up well with any of the oceanographic conditions that we'd measured. And this was surprising and somewhat befuddling, because there had been that pattern of upwelling being a really good predictor of their survival. But I think this indicates that the marine heat wave really changed how we understood the relationship between rockfish and their early survival, and showed that really we needed to link oceanographic conditions to their growth rate. And then if we looked at their growth rate and how that related to their survival, we might get a better understanding of what's going on in their lives. So this next figure I'm going to show you is one of the things where it's kind of validating for everything you've been doing for the last decade of your life, at least for me. And I'm sure many of you have similar experiences in life where you've worked on something for a long time and you've gone out on a limb to try and figure out what you thought would happen. And then you were rewarded with some sort of validation of your existence. And that's what this figure is for me. So here we have rockfish larval growth rate compared to their settlement rate. And this shows that kind of how we predicted, as rockfish growth rate increases, their survival increases to a point. And then there's 2 years where their growth rate was really high, but their survival was really low. And this was kind of an amazing finding. This shows that we can use their growth rate to predict 77% of the variability in their survival. And that when we look at the years when their growth rate was really fast and their survival was low, this indicates the years where the marine heat wave had just started to impact the system and totally threw it out of whack. And what we think happened is that there was really anomalously warm water conditions that was driving really fast growth in larval rockfish. But because the plankton community had changed, very few fish were able to support their really rapid growth, because their prey field had changed. And this meant that only individuals that were successful at finding food were able to survive. And there was really high mortality of all the other fish that couldn't sustain that fast growth. So we found that temperature really drives growth, and that we think that growth rate is the missing link between understanding how oceanographic variability leads to survival variability in rockfish. So when we looked at this over this 7-year time series, we found a really strong relationship between temperature and growth, with faster growth occurring in warmer temperature. And we found that larval growth was a really good predictor of rockfish survival to settlement, and that their survival increased with growth rate until a point, and then there was this unsustainably fast growth that contributed to low survival in the first couple years of the large marine heat wave. And so that information will help us better predict how rockfish will survive and how their populations will fluctuate in the face of changing ocean conditions. So here's one more animation to drive that home. So rockfish growth rate is largely driven by their environment, with slower growth in cooler years. And despite the fact that there is really high quality prey and large amounts of prey available, their survival is low because they're growing so slowly that they're highly vulnerable to predators. And this leads to low survival to settlement in these cooler years. In the moderate years, where their growth is moderately fast, there's plenty of prey available, and that can sustain really rapid or fairly rapid growth rate and leads to high survival to settlement. And then in years where conditions are really warm, survival is low despite their fast growth, because few fish can sustain that growth, and most of them are consumed or they starve. And this leads to low survival to settlement. So one more time, I just want to drive home that growth rate during rockfish larval stages was the link between oceanographic variability and settlement variability, and will hopefully help us better predict how rockfish populations will fluctuate as we move into new ocean conditions. And with that, I would like to thank all the people that helped me with this project, my advisors, Sue Spinagle and Kirsten Grored-Colvert, my collaborators at ODFW's Marine Reserves Program and the Oregon Coast Aquarium, all my lab mates, especially Megan Wilson and Danny Ottman, who were instrumental in helping with the SMRF program, and also Kelsey, Keely, Miriam, Jamie, and Margaret, who are just amazing people to work with. My funding sources from Oregon Sea Grant, Hatfield Marine Science Center, and ODFW's Marine Reserve Scholarship. And with that, I would love to take any questions you all have. Thank you for your time.
Awesome research, Will. Great presentation. So if you have questions, please feel free to add them to the Q&A in the chat. And I always have one I like to start out with. And I always am curious about what was your inspiration or an aha moment or an experience that made you kind of want to go in the direction of doing this research or learning more about, the growth of fish?
That's a super interesting question. I think kind of the end of my master's is what set me on this track. So I looked at how ocean acidification impacted behavior and swimming ability of a variety of species of rockfish, and found that different species that had different real life history strategies responded differently to ocean acidification. And that led me to think, okay, well, what's really happening during their early life stages? I want to learn more about that and see how the environment interacts with their early life stage to affect these fish populations. So that's what was the major question I had after my master's. And I talked with my master's advisor and was like, how do I go about answering this? And he said, you need to go work with Sue Spinagle at Oregon State University, because she is the expert in doing this, and she's just moved to the West Coast. And I was like, all right, then that's what I'll have to do then. And I'm really glad that I did.
Very cool. Nice. Have you discussed this research with the Oregon Murrelet Project, as the murrelet feeds on rockfish, juvenile rockfish, and their nesting success is linked to ocean productivity?
Great question. I have not directly interacted with them, but I believe there is strong collaboration between seabird monitoring and investigations of fish populations in the California current. So I know there's a lot of work between many seabird groups and NOAA scientists who monitor juvenile life stages of fishes, but I have not directly been involved with conversations with the seabird people, no.
And it looks like your presentation was really thorough. We don't have too many questions coming in right now. So with that, I think that we can end the presentation. We got some notes here. Great talk. Thank you for the presentation. And so just to remind everybody, we did record the presentation. So this will be available for future use or learning. It's really great research and information to share with all these other groups if you're participating in any of these projects. Oh, somebody mentions here, can we hear a little bit about the salmon?
Sure, if you don't mind, like a 10-minute.
Yeah, no, go
swap.
for it.
Okay, let me stop sharing my screen and I'll get a little salmon thing going for you. And also, if you think of any other questions, my email is fennieh@oregonstate.edu.
And I will put that in the chat too
Cool.
for folks to copy if they'd like.
And I just want to make sure people know that the salmon work is still rockfish related. Let's see. Okay, you seeing a new slide?
Yep, we see your new slide.
Cool. Okay, so this project focused on looking at predation of pelagic juvenile rockfish during their transition from their offshore to nearshore life stage, and involved collecting rockfish simultaneously with salmon, which are, I believe it was juvenile coho salmon as well as Chinook salmon that we were collecting, and juvenile coho salmon are major predators on juvenile rockfish. And I'm just going to go through this one a little quickly to get you to the cool parts. So just a little bit of background is that coho salmon are highly piscivorous as they enter their ocean environment. I believe that at their smolt stage. And this figure from some of Elizabeth Daly's work shows how as they enter the ocean and start to grow, the proportion of their diet that is rockfish increases and is about 25% of their diet during their early ocean experience. And they're actually an important predictor, rockfish are an important predictor of coho salmon survival. So it's kind of cool to work with both salmon and rockfish. I know these are probably people's favorite fishes on the Oregon coast. And based on those same growth mortality hypotheses, we predicted that juvenile coho salmon would be selective predators on juvenile rockfish and consume the smaller, slower growing individuals, and there would be higher survival of larger, faster growing rockfish. To do this, I participated in some of NOAA's juvenile salmon ocean ecosystem surveys, particularly in 2018. And we were able to simultaneously collect juvenile coho salmon and juvenile rockfish in the same net. And then we compared the traits of rockfish that were alive when we caught them to rockfish we dissected out of the stomachs of coho salmon, and compared the otolith characteristics between these two groups to see if there's anything different between the survivors and those that were consumed. And here's just a quick image of what consumed rockfish look like in the stomachs of salmon. They're pretty highly dissolved or digested, and to identify them, we actually had to do some genetic work to identify them to species. And we did similar otolith microstructure analysis. And the major difference I want to bring to your attention is we looked at their recent growth rate. So this is their mean growth over the last 5 days of their life to see exactly how their characteristics immediately prior to encountering the salmon were predictors of their survival. And again, we thought that survivors would be larger and faster growing than the consumed fish. In the next couple plots, survivals will be in green, and you can think of that having them having a green light to pass by the salmon, and the consumed fish are blue because you're sad that they were eaten. So here we compared size of consumed versus surviving rockfish, and we found that the survivors were about 15% larger than the consumed fish. Next, we looked at their age, and somewhat surprisingly, we found that the consumed fish were younger than the survivor fish. And this kind of caused us to scratch our heads a little bit. We then looked at their recent growth rate and found that the survivors were growing considerably faster than the consumed fish. So that was kind of what we expected. But that age thing was really interesting, and that inspired us to look at their entire growth history. And we found that, somewhat surprisingly, in their early part of their life, surviving rockfish were growing slower. There are these green dots down here, and they're below all these blue dots for the first 30 or so days of life. And this indicates that actually during their larval period, the survivors were growing considerably slower than the consumed fish. But by about day 30, that pattern switched, and then the survivors were growing faster than the consumed fish. And this is kind of what they were doing immediately prior to encountering the salmon. And this figure just shows how that change in growth actually led to larger size at age among the blue consumed fish compared to the green survivors through the first 30 or so days of their life. And then that pattern starts to switch as the faster growth rate of the survivors leads to larger size. And as you may recall from that last presentation I gave, sea surface temperature played a major role in the growth rate of these quillback rockfish, with warmer water temperature contributing to faster growth rate. And when we looked at when the fish were born compared to the sea surface temperature that they experienced, we found that in general, the survivors were born slightly earlier in the season than the consumed fish. And the consumed fish, for the most part, were born later when waters were warmer, and this might contribute to their faster early growth. So really quickly, what we found was that survivors were born earlier and developed in cooler water than the consumed fish. Consumed fish were born later, developed in warmer water, and grew faster early on. However, the survivors actually caught up and were growing faster and were larger in size immediately prior to encountering the salmon. And the salmon, this led to variability in their traits. And the salmon selected against the younger, smaller, and slower growing individuals. And this led to, I guess this really followed what we expected based on the growth mortality hypothesis. And the major differences between the slower growing, smaller consumed fish and the faster growers might actually have to do with the condition of the mothers that produce these fish, where older, larger females are thought to produce higher quality larvae because they have higher lipid reserves and can actually give birth to larvae earlier in the year, which may provide a survival advantage as it gives them more time to grow and develop before they encounter these salmon. So potentially these survivors were actually, their survival was predicted by who their mothers were because of the traits that they possessed. So that was something that I found really interesting. Oops, and this is just a recap of some other things. So yeah, temperature is a major part of driving variability in rockfish early life history traits, and salmon actually are selected predators on rockfish based on what kind of traits they have. So it was really cool to see that work out.
Yeah. So one more question trickled in.
Sure.
Can you speak about the Hasita Bank and connectivity to the nearshore?
I can try. I don't know as much about that area. Most of my work is on nearshore rocky reefs, and I've done a lot of monitoring of the nearshore marine reserves. But I think one of the cool things about Cedar Bank is that it's this kind of hard to access habitat that is really good for rockfish. And actually on some of the NOAA cruises I've been on, if they have time during the day, they'll go fishing off there during their downtime because they know it's a really good fishing spot. And so that might be a really important de facto reserve, just because it's so hard to get to, that the fish can get really large and really old. And that might play an important role in determining survival of the young fish. Because if these quillback rockfish were born from older mothers and they had higher survival, at least in terms of encountering salmon, that may also hold true for other predators and for survival in general.
Very nice. And then just a quick note here that says, thank you for the 2 for the price of 1 presentations and the seamless transition. So thank you, Will, for taking the extra time to share the salmon bit as well.
Yeah, it's really fun to talk with this to an audience that's interested. I'm really glad.
It is so fascinating, the research. And I remember when I learned how you could tell the age by the otoliths and everything. It's truly fascinating what we can learn about the fish, about rockfish.
Yeah, could I ask for anybody who didn't see Megan Wilson's presentation on cabezon, could we post a link in the chat to that YouTube video? So if anyone else wants to see more information on fish early life history and otolith work, that they can check that out as well.
Yes, you bet. I will go ahead and do that. In just a second here, I'm going to find it. And in case anybody's interested in any other recordings, we have posted them on our blog. So if you go to our website and go to our blog, you'll find them all there. This is the link here that I've just put in the chat for the one Megan Wilson give about the cabezon. And then also on our YouTube channel, they reside. So definitely check it out. Fascinating research. I'm so grateful that Kristen from ODFW referred you guys to me for this presentation and speaker series. So again, thank you so much, Will, for participating today. I'm really grateful to have you. Other thank yous coming through the chat and how you explained a complicated subject really well. So I appreciate that. It's always nice for us common folk to get it in an understandable way. And with that, I hope you all have a wonderful weekend and hope to see you at a future presentation. Have a great day.
Have a great weekend, everyone. Thank you again for the invite.
Yeah, bye-bye.